Here, we will cover how to upload your FTMS data to FREDA. We will begin in the Multi-omics application portal. First, we will navigate to the Data Upload tab. We will select FTMS for our data type and give it a project name. We'll be using the same example data that was downloaded in the previous video. As covered in the data requirements video, there are two files. The first is the expression data file, which we'll upload by clicking the Browse button under expression data. The second is the biomolecule information data. As we can see, a table summary appears to the right for both data types, and where we can verify the unique mass identifier column is the same for each file. Click Save Project and Submit to Server. We can verify our project is uploaded in the Manage Files tab, then navigate to the Map Store. We will select FREDA, select our project from the drop-down menu, then launch the app. We will be greeted with a message where we will click Go to Upload tab. From here, we will select our mass column as the IDs.
And as you can see, a table summary appears here and shows that it has the same unique mass identifier column. Now we're prompted to select whether a molecular identification file has formulas or elemental columns. In this case, we have elemental counts for carbon, hydrogen, oxygen, nitrogen, sulfur, and phosphorus. We select elemental counts and are then prompted to select which columns identify those counts. Carbon and hydrogen are automatically detected, and the rest can be added with the input widgets here. You'll be asked whether or not your molecular identification file has information on isotopic peaks. In this case, we select yes, and that we do want to filter these peaks. You're then prompted to select which column contains this information. In our case, it's the column C13, and a 1 indicates the presence of this data. Once you've filled out all this information, you're ready to create the data object that will be used in FREDA. Click Process Data. The object is now created, and you're prompted to either review this page or continue to subsequent tabs. There are summaries on the numbers of peaks in your data, the number of samples, and the number of peaks with identified formulas. Your data is now successfully uploaded. In future videos, we'll go over how to perform further preprocessing steps such as grouping and adding metadata.